This chapter begins our exploration of inheritance. And so we're going to start by looking at the molecular basis of inheritance. And so we first need to understand what the genetic material or the molecules of inheritance would need to be able to do. And so there's really four key things that the genetic material or criteria that the genetic material has to meet. It has to contain the information needed to make a whole organism. So it's got to have enough information to make a whole human or a whole monkey or a whole dog. It's got to pass from parent to offspring, right? We know that offspring look like the parents and that information is coming from the parent. It also has to be able to pass from cell to cell. So we know that every cell in your body is getting genetic material passed to it from the very first cell that you started as, the fertilized egg. The third criteria is that it has to be accurately copied. So again, if you're starting with one cell, the fertilized egg, and going to your whole body, which is 50 trillion cells, each one of those had to be accurately copied from that very first cell. And then the last criteria is that it needs to be able to explain variation between individuals like you and me, and also between species, like between humans and chimpanzees and cats and dogs. So in the late 1800s, scientists realized that there would have to be a biochemical basis to inheritance, and what that means is that there's a chemical molecule that's responsible for inheritance. Why is that such a leap in science? Well, up until the late 1800s, there was a lot of other theories about inheritance, things like ether and gas or spirit that passed from one thing to another that carried the information. But by the late 1800s, scientists were really pretty sure that there had to be an actual physical molecule. And they quickly decided that it was probably in the chromosomes. Part of the reason why was that chromosomes were something that they could see even with the very first earliest microscopes. And they could see that the chromosomes got divided up when cells were dividing. And this is something you'll be able to see when we get to chapter 15 too. So they could actually see the chromosomes go from one cell to the other cell, which is one of the criteria for life. Now the issue that remained for them was that chromosomes have both protein and DNA in them. So two different molecules. And so they were left with the question of which one was it? Was it the protein in the chromosomes or was it the DNA in the chromosomes? And for a lot of years, and through the first part of the 1900s, most scientists thought that it was going to be the protein part of the chromosomes. And if you think about this, why would they have made this hypothesis? Well, remember back to chapter 3. Proteins are made out of 20 different amino acids, whereas DNA is only made out of 4 different nucleotides. So if you're looking for something that can explain variation, explain differences between individuals and differences between species, it kind of seems like 20 different amino acids would get you more unique combinations than 4 different nucleotides. And so that was kind of one of the reasons why they really favored proteins. But of course, what we know now is that DNA is definitely the genetic material of all living things. A whole series of experiments showed that DNA, not RNA or protein, determines traits in individuals. And finally, in the 1950s, and this was kind of the capstone of this whole series of experiments, was the modeling of the three-dimensional structure of DNA that we're going to talk about, called the double helix. And that made it very clear how DNA would work as it was being copied. And then over the last 50 years, we've really clearly now understand how DNA actually leads to traits. So again, it's a relatively new theory, but it's very, very, very well established now. And in fact, it's very widely accepted. And if you think about all this crime scene evidence and all this stuff, that all hinges on the DNA theory. So just a reminder from chapter 1 that when we use the word theory in science, we're actually saying this is something that we feel very strongly about and we believe to be true to all of our knowledge today. So DNA is a very complex molecule. It starts with nucleotides. So individual nucleotides, like these, are the basic monomers. So DNA is a polymer, and the nucleotides are the monomers, again, sort of referencing back to chapter 3. So we talked about beads a lot then. So Each bead is a nucleotide and it forms a long polymer. And in the case of DNA, that's through phosphodiester bonds. So each one of those nucleotides are connected through a phosphodiester bond. So this will form a strand of DNA or RNA. So this also holds true for RNA. In DNA and DNA only, two strands, so I have two strands, they're going to wrap around each other. And I'm not a very good drawer, as you've seen in class. But the two strands are going to wrap around each other into what's called a double helix. And here's an image that's showing you the really pretty double helix drawing. And then in living cells, the double helix will further wrap around proteins to form chromosomes. So it's going to wrap up even tighter and form chromosomes. And some of you may have seen images of these before. This is a duplicated chromosome that we'll be talking a lot about in the mitosis chapter. And then each organism has a genome, which is multiple chromosomes. And that includes all of the chromosomes that are needed to make that organism. So for example, in humans, we have 46 total chromosomes that make up our genome. So every single human has 46 chromosomes that make up their genome, and that's the full complement of genetic material needed to make a human. So we're going to go through the structure of DNA and RNA here. And some of this is review, some of it will be new. So DNA nucleotides have three components. So just a really basic drawing of a nucleotide is the sugar, the phosphate, and the base. So that's the three components. In the case of DNA, this is a deoxyribose sugar. And the difference is here. So deoxy means less oxygen, right? So in RNA, we'll see that there's an oxygen there. So that's the difference. And then this part is the variable part. So the base is variable. In that position, it either has an adenine, a thymine, a guanine, or a cytosine. And so we'll generally refer to nucleotides as A, G, C, or T based on the base that's present, but always remembering that there's also the sugar and the phosphate. The bases are grouped into two families. They're either purines or pyrimidines. And the way that I remember which one is which is a little mnemonic that I learned. Pure as double gold. So the purines are A and G, and they have a double ring. So notice there's one ring, two ring, one ring, two ring. So the purines have a double ring structure. The pyrimidines are the other ones, T and C, and they have a single ring structure. So RNA nucleotides are really similar. They have a phosphate group, a sugar, and a base. The difference is again in this position. So remember I said deoxy means less oxygen. This one has an oxygen in that position as well. And so this is a ribose sugar. So RNA ribose. The R is for ribose, and in DNA the D is for deoxyribose. The bases in RNA are also mostly the same. So there is A and G in both in RNA, and then there's cytosine in RNA, but instead of T, you'll find a U in RNA. So instead of the T. So in RNA and DNA, there's a standard numbering system that helps scientists communicate about these molecules. So they will always call this nitrogen the 1 position, this is the oxygens in the 2 position, this nitrogen in the 3 position. So it's just a way to say where an atom is located. And the one that we're most concerned about is the sugar. Because the numbering is different here and here, right, but it starts back at 1, this is called the 1' position. So we call that prime, 1', 2' position, 3' position, 4' position, and the 5' carbon. So the ones that were going to be especially important to us in this chapter and the next chapter is the 3' carbon and the 5' carbon. The reason why those are going to be important is because that's going to be how the strand forms through the 5' and 3' carbons. So just write this up here. The most important ones for you to understand are the 5' and 3' positions. And again, the reason why that's so important is that's how the strand is going to form. So if I look at this strand of DNA here, this is a 5' carbon here, and this is a 3' carbon. So the bond between nucleotide 1 and nucleotide 2 is between the 3' carbon of the first nucleotide and the 5' carbon of the second nucleotide. So the 3' carbon of one nucleotide and the 5' of the second nucleotide. That's where the bond forms. Remember, that's called a phosphodiester bond. Okay, so what that means when I look at this whole molecule is that if I look up here, the top of the molecule in this picture is what's called a 5' end, or to help you remember this, a 5' free end. So notice that here's the 5' carbon, there's nothing else attached to it. If I look down at this side of the molecule, this is the 3' carbon. The 5' carbon has another nucleotide attached to it, so it's not free. So this end is a 3' free end. There's nothing attached to the 3' free end. So DNA and RNA molecules always have directionality. One side of the molecule is the 5' end, the other side is the 3' end. And you need to always know which end you're talking about because the molecule is flippable. So let me get out of pen mode here. Not letting me get out of pen mode. If you imagine rotating this molecule, so let me rotate this for you. Right? What was on top will now be on bottom. And let me just move it aside here so we can see it. And so now that I've turned the molecule, this 3' end is now on top, and the 5' end is now on the bottom. Remember, earlier it was the opposite. It's the same exact molecule. I've just flipped it around. And remember, that is not an isomer, it's not a difference, it's the same molecule just flipped over. So it's always important for us to know what the orientation is. So whenever you're writing DNA and RNA sequences, you should always write them like this. There should always be a 5' and a 3' indicated, because I could also write for the same exact molecule, I could write it this way instead. So these are the exact same thing, just written in a different way. The three-dimensional shape of DNA, and this is for DNA only, okay? RNA is not going to do this. DNA takes on something called the double helix. The double is referring to it having 2 strands, and the helix is referring to a helical shape, so kind of like a spiral staircase. So the 2 strands will wrap around each other so that the sugars and phosphates are on the outside. And that's the sugar-phosphate backbone. So the red here is the sugar-phosphate backbone. And they will, here's one strand, here's a second strand, it's on the outside. So if I imagine drawing this just a different way, 1 strand, 2 strand, they're going to be oriented slightly differently so that the bases face in. So in one strand, the base is facing to the right, and the other strand, the base has to be facing to the left, so that the sugar-phosphate backbones are on the outside, which is these lines here. So the reason why it's going to be stable like this is that the bases are going to bond to each other on the inside. So if this is an A and this is a T base, they will form hydrogen bonds. So hydrogen bonding is going to hold these together. There's very specific rules. An A hydrogen bonds to a T, or a T to an A. And G's and C's hydrogen bond together. And that will be, and that's in this figure, see these little lines here? Those are the hydrogen bonds between the bases. So this is another image of the double helix. So you have your sugar phosphate backbone. So notice phosphate, sugar, phosphate, sugar, phosphate, sugar, and then on the other side, phosphate, sugar, phosphate, sugar, phosphate, sugar. And then in the center, you've got your bases hydrogen bonded. A's and T's form 2 hydrogen bonds. So A has two hydrogen bonds between it. G's and C's have 3 hydrogen bonds. So that's why you'll see 3 lines here. So A to T and G to C is important to the double helix because that's always keeping the width consistent. So remember that the purines are the double ringed bases. So there's adenine, there's guanine. They always have the double rings, whereas the pyrimidines always have single rings. So as long as you have a purine to a pyrimidine, so base pairing is always purine to pyrimidine, then you're always having 1, 2, 3 rings across. So that keeps, so the A to T or G to C is always 3 rings across. And so if you imagine touching the double helix, it would always be the same width. Because of the base pair rules, the 2 strands are complementary. So that means if I know the sequence of one strand, like if it was A, G, C, C, A, for example, then the other strand must be the matching base pairs. So T, C, G, G, T. So notice A to T, G to C, C to G, C to G, A to T. So it has to have the opposite of the pair. And that's what's called being complementary. So once you know one strand, you can alwa' figure out what the other strand is. The strands are also anti-parallel. That means they have to run in opposite directions. So remember we said earlier that there's 5' and 3' ends. So they have to have opposite ends. If this is the 5' end on this strand, the matching strand, that must be the 3' end. And if this is the 3' end of the first strand, the matching strand must have the 5' end in that position. So if we look at this diagram, notice this is a 5' end, and this is a 3' end. So I see that free 3' end and a free 5' end. And if you look at the sugars, see the oxygen up top here? The oxygen is pointing up. So I've just drawn that a little bit bigger. If you look at this molecule, so the matching strand, this is the 3' end. So notice here's the free 3' carbon. And if I look down here, here's the 5' carbon. So this is the 5' end. And if you look at the oxygens are pointing down. So I always think of them as little arrows. They are actually different shapes. Here the oxygen points up like an arrow. Here the oxygen points down like an arrow. So they're running in opposite directions. So now that you know these rules, if one DNA strand is 5' CAT, GCA, TCG-3', you should be able to figure out the complementary strand. So another tool that knowing the base pair rules gives us is the ability to figure out the ratios of nucleotides. So if cytosine is equal to 38% of the nucleotides in a cell, since C base pairs to G, we know that for every C we must also have a matching G. So that means G must also equal 38% of the nucleotides in an organism. So C and G together take up 38 times 2, which is 76, right? So that's C and G. So that only leaves 24% for A and T together, and A and T must also be equal. So we know that 24%, divided by 2 is equal to 12%. So A is equal to 12%, T is equal to 12%. So another way to sort of put this is that A and T must be equal to each other, C and G must be equal to each other, and all 4 of the values must add up to 100%.